Okay, I'm back again. Battery was running flat. All right, so let's go back to here now. Okay, so I've explained the simple cell cycle on page 53, but now, okay, if you go and look on the internet, you're going to see more complex ones, all right? And we do not need to know all of this, okay? So they will talk about the blue arrows here on the outside, that represents interphase, okay? So that's when the cell's undergoing normal processes or it starts preparing for cell division, and they give it these names like G1, S, and G2. Please, if you ever see that, don't worry about it. Then actual mitosis over here, so prophase, metaphase, anaphase, and telophase. I think they've got too many little, oh, they have early prophase, late prophase, all of that, okay? Because I thought there were too many of these over here. We simplify it. And then you've got the actual division of the cytoplasm, and then you end up with 2 daughter cells, and those cells can then, okay, they can also undergo cell division. So think of cells in your skin or lining your esophagus, etc. Or, you know, if something's wrong with the cell, then it stops, you know, it dies. Or, for example, the cells to form, you know, that break down to form our fingers. Okay, so don't get bogged down if you see things like that. All right, so now we're going to move, we're just going to revise again what happens to the DNA before we look at cell division. Okay, so this diagram shows a cell, there's the nucleus, and you can see that this cell is ready for cell division because there are the chromosomes, and they've got those 2 DNA molecules, okay, joined by a centromere. So there was 1 DNA molecule, and it opened up, and then you ended up with 2, it copied itself, and you ended up with 2 DNA molecules joined by a centromere. Now, it's quite difficult to understand how DNA fits into the nucleus of a cell. In fact, if we looked at the 46 chromosomes or the 23 pairs of chromosomes in human cells, and you unraveled them and put them end to end, you can see here they're showing a diagram of unraveling the DNA molecule, and it's actually wrapped around these little blobs here. Those are histones. So we learn about that in grade 12. It's kind of like the glue that's holding that DNA molecule in that very tightly coiled helix. All right, and you can see it's unraveling more and more and more. There's, you know, very simple helix, and there's the DNA molecule all stretched out. All right, so if you had to put all 46 chromosomes end to end, believe it or not, that, you know, I'm 1.59 centimeters, but that the DNA in every single cell in your body, okay, diploid cells, is actually, if you put it end to end, it's 2 meters long. That's a lot of DNA. So how does so much DNA fit into a tiny little cell that we can't even see with the naked eye? It's all about supercoiling. So coiling upon coils, like when you twist an elastic band and it twists and twists and twists and twists, just like they've shown here, and how it wraps around histone proteins into a very, very tightly coiled molecule. All right, so 2 meters of DNA in every single cell, 46 chromosomes coiled up inside the nucleus of every single diploid cell. Obviously, in gametes, there would be half, you know, 1 meter of DNA. All right, now, in grade 12, we learn what chromosomes do. So over here, there's a very simplified representation of a chromosome which hasn't replicated, and these little regions that they've drawn, well, it's regions of the DNA called the genes, and the genes control what the cell does, okay, which I've mentioned before. So red blood cells make hemoglobin, white blood cells make antibodies, etc. Genes determine the shape of your nose, your ears, your skin color, your toe length. Oh, there's so many different things. And we look at that in grade 12 when you do genetics. Okay, so there's the 2 DNA molecules. If the chromosome replicates, there's the 2 DNA molecules, and they're held together at a region called the centromere. The 2 tightly coiled DNA molecules held at a region called the centromere, and those 2 DNA molecules are copies of each other, and they're known as chromatids held together at the centromere. All right. So they've indicated here that there are, 6 or 7 genes on this chromosome, but in actual fact, you know, we've got 20,000 genes spread out along all the different chromosomes. So 1% of those genes makes us different to each other. I've got dark hair, someone's got, you know, reddish hair, or blonde hair, or I've got very straight hair, someone's got curly hair. You know, I've got quite a dark skin, someone's got a paler skin, or a more fair skin, or, you know, a dark skin. We differ in 1% of our genes only. Okay, so what do the rest of the genes do? What do the other 99% of 20,000 genes do? Well, we all have a pancreas, we all have fingers and toes and stomach and brain cells, and we all have a heart that pumps, and that's the 99% of us that is the same. And that's what I always find so amazing. You know, we look at eyes, we look at hair, we look at shapes of faces and things. And yes, we're all different, okay? And it's 1% that makes us different. And that's something that we really, really need to embrace. Okay, we are 1% different. I think that is really biologically important. Okay, so how does DNA replication occur? So remember, the DNA has to replicate before the cell divides. So this is what we do in grade 12. But I think, you know, in order to understand the process, and it is shown a little bit on page 53 of your book, you know, but it's very vague. So here's the original DNA molecule. You take that DNA helix and you stretch it out. So now it's in the shape of a ladder. And there, adenine joined to thymine and cytosine to guanine. Okay, we're not going to ask you that. But what happens is that the DNA molecule splits. There's an enzyme which causes it to split. And then new little nucleotides, these little red ones, are brought in. And they join onto both sides. And they join on exactly the same way. So you end up with two DNA molecules there. And it's half old, half new nucleotides. Half new, half old. And there they are, still joined by the centromere. The two DNA molecules still joined by a centromere. That was a chromosome, which has now become a replicated chromosome. And each of those DNA molecules are called chromatids. All right? Sister chromatids, in fact. Okay. So there's another diagram. Okay. If you go on the internet, you're going to find lots of different representations. You know, sometimes they're drawn and it looks like this is right in the middle, you know, but it's two chromatids joined by a region called the centromere. It's not even a black blob like that over there. This is what a DNA molecule, or at least a chromosome, looks like in real life. Very highly magnified with an electron micrograph, coiled upon coiled upon coiled. All right? These are the actual, that's part of the DNA molecule. All right? So it's very, very long, very, very thin. And because it's so thin, 2 meters of DNA can fit into a cell. Okay, and there's one chromosome. There are 46 in each of your diploid cells. All right, now, we've got 46 chromosomes per cell, and that's what they would look like. Okay, so once they've all replicated, there they are. You can see they've got different lengths. Some of them are long, some of them are very short, like that one over there. Some of them, the centromere is more in the middle. Some of it is more near the end. Okay, that's how we can distinguish them. So you can actually pair them up. So there's chromosome 1, there's another chromosome 1. So that chromosome there on the left is actually 2 chromatids, but they're so close to one another that it looks like one. In other photos, you can actually see the 2 chromatids. This is the other copy of number 1. So the number 1 came from the egg, and the other chromosome 1 came from the sperm. So that's why our cells are diploid. All right, so each of these here is actually 2 chromatids joined. They're quite close to one another. They're not shown separate as in this diagram over here. They're stuck together. So there's chromosome pair 1, chromosome pair 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23. Now, they actually do take a photograph of the chromosomes when they look like that, but then they stain them and they get this little banding pattern. And then there are, you know, people are trained, geneticists and that are trained to actually match them up according to the length and where the centromeres are and the little staining pattern. And then they can determine, for example, if there is a gene that, you know, there's something wrong with it. They can actually determine if the person has a gene for breast cancer or a gene for, you know, getting Parkinson's when you're older, etc., or cystic fibrosis. They can actually map our genes, and that's what they're trying to do, is figure out what all of these genes do, and to be able to look for, you know, ones that have mutated that can cause disorders or disease. All right. Now, this kind of diagram is actually known as a karyotype. So I've said interest here, but it just kind of ties in with grade 12. So it's an ordered photograph of the pairs of chromosomes from pair number 1, all the way down to number 23. Now, the 23rd pair, that's the sex chromosome. So this would be the karyotype from a boy because there's a long X and a short little Y. So in a female's karyotype, there would be 2 long X chromosomes, female. And in lions, for example, it's actually the other way around. Okay, so it differs in different species. So that's what a karyotype is. Picture of the chromosomes ordered in their pairs. All right. So this is now another little summary of what happens in cell division. You can match it up with the one that I explained with the circles in the previous lesson. The interphase, the cell prepares for cell division, the chromosomes replicate. Then you get nuclear division. It's called karyokinesis, center of the cell splitting. That's where the chromosomes separate into daughter cells. So IPMAT. Okay, there it is. Interphase, normal cell processes, and prepare. Then the nucleus divides, or the chromosomes are divided between daughter cells. Prophase, metaphase, anaphase, and telophase. And then after that is cytokinesis, where the cytoplasm actually splits. And if you remember, in the next video that you're going to watch, you're going to see what happens in plant cells. All right. So this is a diagram to show mitosis, but I'm going to let you watch the video of where I actually draw on a diagram to explain what is happening at each stage.